Well, I guess I'm trying sometime, hopefully maybe today, later today or tomorrow, I will send what we call the topics for your essay, okay, which is really needed at the end of the semester. Okay, so basically we'll do that, I guess. Do you see this clearly?
Yes.
Okay, that's what to know. Okay, thank you. Basically, as already you know, with the introduction material, I basically cover some introduction about fungi, okay? But also we talk about, mainly we talk about their importance, and basically we talk also about the general characteristic of the fungi. And in this one, as a matter of fact, I cover these points. Let me just, okay, make sure that okay. Basically under the biology and characteristic of fungi, I cover these points. I mentioned to you that most of fungi are composed of hyphae. Fungi are heterotrophic absorber. Then we talk about fungi have unique variation in mitosis and meiosis. And also we mentioned that fungi reproduce both asexually and sexually. And finally, we talk about basically the evolution of fungi, okay? And basically, by this one, I end this one because I finished the review last lecture. And simply, there's not much, but I guess just hopefully if I can see it here. Yeah. It looks like something like this here. It's not, I don't know, this way is better or that way is yeah. And basically that basically comes from the aquatic. So basically we have, as I mentioned to you, the chytrids, okay? The chytrids always remember what you call the aquatic fungi. Though I'm going to start with you basically with the slime mold. And then we have the zygote fungi, or basically these are known as the zygomycetes or the Zygomycotina, the zygote. And then we have the sac fungi, which also I give it to you known as the Ascomycotina, and the last one, which is commonly known as the club fungi, which include the mushroom, or we call them as the Basidiomycotina or Basidiomycetes. And of course, we have the group already I mentioned to you, which is basically the group known as the Deterio Microtina or the Deteriomycetes. So I put it down for you to look like here, okay? Of course, okay. Now, the major group of fungi, the Myxomycotina, which is the slime mold, okay, the Mastigomycota, this is what called the zoospore forming fungi, or basically these are the aquatic fungi, because they form spore with the flagella called the zoospore, and we're going to find there are two groups. Some group, they will make a flagella with, spore with one flagellum, other, they may make zoospore with two flagella. We'll talk about this. And then, of course, we have called the Zygomycotina, or we call the Zygomycetes, and which is known as the zygote fungi, because they, as I mentioned to you, all the grouping based on the spores are producing during their sexual production, not at the asexual, okay? And then, so that these, they produce the zygospore, and we call them Zygomycotina or the class as Zygomycetes. And these are known as the sac fungi, or we call them the Ascomycotina or the Ascomycetes. And you have the Basidiomycotina or the club fungi, or we call them the Basidiomycetes. And as I mentioned, the last group here known as the Deterio Microtina. These also known as as I mentioned, known as fungi imperfecti. Okay, because very simply their life cycle is not perfect. Mostly they have what you call the asexual life cycle. So either the sexual life cycle is not yet being discovered or does not exist. But it seems most of these fungi, when they find their sexual life cycle, most of them mostly belong to Ascomycete later on or Basidiomycete. They put them in the group. But also this is a very large group. So which are the group I'm going to start first today with the Myxomycotina or Myxomycota, or known as the slime mold. Okay.
Excuse me, Dr. Nasrullah.
Yes.
You're not sharing the projector onto the screen.
I'm not sharing the projector.
Yeah, we can't see your paper like you normally show.
There's another screen.
You see it?
You just got to pin the other screen. He's showing it.
Sorry.
So you don't see this?
Yeah, it's all right.
It's okay now?
Yes, we see it.
Okay. So I don't know really. I didn't do anything different. It's still the same thing. I don't know. This connection, how it works out. Okay. Yeah. So, but if you didn't see, let me know. Okay. But now you see it.
Yes, definitely.
That's for sure. Okay then. Now, basically, as I mentioned, okay, now we're going to start here. Hopefully, I'm going to put some, okay. We have here the okay. Well, this is something here really I just try to see what's going to work on this one. Because always there's like some sort of delay here sometimes. Okay. We call them Myxomycota, slime molds. Okay, good here. Okay. Or as a class, call them Mycetes. Okay, known as Myxomycetes, or sometimes we call them this doesn't matter which way. Okay. Yeah, I tried to put all the classification in the beginning so that okay. We have a class Myxomycetes, and we have the other class known as can you read this? Called Acresiomycetes. Can you read it?
No.
No?
You should need to zoom out a little bit.
See it now?
Yep.
Yeah.
Okay, good. All right. Now, Yeah, because basically seems the exposure area is not that. Now, usually the myxomycete or the maxomycete, this one here, known as commonly, this group here known as Plasmodial slime molds. Can you see this? Okay. Before that, this one here, the class maxomycetes known as Plasmodial slime mold. This one, the acresiomycetes, known as commonly as what they call cellular slime molds. These are basically the two main classes. Did you get the lab for the slime mold? Do you have the lab now?
Yes, we have access to the lab now.
Yeah, so it's become more easier, yes, okay. And, okay. But I'm going to give you another class with this one here, known as Plasmodiophoramycetes. Called Plasmodiophoramycetes. Can you read it? Okay. And basically these are, they can cause parasite, okay? These are known endoparasitic slime mold. Okay. Now, did you follow this? Good. The best example, I'm going to example on each one here. Now, the best example on this one here, the Plasmodial slime mold, I'm going to put because there is no room here. The best example of species here for this one is known as Physarum polycephalum. Okay. This is the most typical example about what you call the Plasmodial slime mold. Okay. But unfortunately, I don't know if they showed you a picture dish in there. Okay. It's not that easy to see here, but anyway. Okay. Now.
Can you shift the paper up a little bit?
Say that again.
Can you shift the paper up a little bit? Oh, thank you.
Can you see? Yeah, because I make sure this is example of the Plasmodial slime mold. Okay. The first one, myxomycetes. Now, the best example on the acresiomycetes or the cellular slime mold, okay, is known as Diptostelium discodium. On the cellular slime mold called Diptostelium. I'm going to repeat it all again, but just at the moment so that you can put it in the beginning here. Sorry. Diptostelium discodium. Always remember when you write a Latin name or the scientific name of a species, always remember, of course, consists of two names. Okay, this is known as the generic name. This is a specific. Okay, but two names, and always the generic name always will start with capital. Note the specific. Always remember that. Of course, I'm not mentioning the rest because and I underline it because always the scientific name or the Latin name should be written in italic. But if you write it, then in that case, all you underline, just for your information. You got this? So basically, the example we're going to use for Fusarium polycephalum as example of the Plasmodial slime mold, and Dictyostelium discoideum example on the cellular slime mold. And I think the other one, there are quite a few later on we're going to mention in the Plasmodial formicid. But these are the two typical examples we're going to find here. Now, first, I'm going to start with you, as a matter of fact, a little bit of a general characteristic of the group. Okay. The general characteristic, that means the whole group of the slime mold, what distinguishes them. The slime mold, as a matter of fact, as I mentioned to you here, okay, these are sometimes known as we call them sometimes the fungus animal. Okay, just give me a second here. See now. I don't know, maybe they show a picture in the lab, but basically that's how they look like. Sometimes they are plasmodial like this. This is basically, as you can see, is a plasmodium that creeping. Okay, and this is at the back picture here. You can see the tree or something, especially in the moist condition. And some of them, the Plasmodium slime mold is the most clearer one, or the most obvious in nature. It creep, and basically because in its vegetative state, basically it feeds on a small microorganism, bacteria, and others. But later on, when they, as I mentioned, when they try to produce their spore is similar to the fungus spore. Okay, those, some of them, they produce quite a few nice type of spores. Okay, and okay, so basically like this. And if I have some transparency, they look like this one here. Okay, do you see it? So this is an example of the one I told you, Fusarium polycephalum. You can see it has veins, and unfortunately, of course, at the moment you can not see it in the lab, because in that case you don't see cell are equal. So that's why the structure is all multinucleate when it's cell there, and it has veins. And really, if it was a live thing, you can see that in the veins under the microscope, very easily you can see what's called shutter streaming of the cytoplasm moving in one direction or two directions. Very easy, you can see this one. Okay, but as I said, when they produce their spores, sometimes like this one here, they produce different spores, and really their spores is very nice. They look different shape like this one here. Okay. And of course, later I'm going to give you a detail of their labeling. I don't know if they give it to you in the lab, but they have, so basically when they produce the spore in the end, it looks what they call very nice. So these are the Plasmodial slime mold, or the general about the slime mold. So we're going to talk first in general here about their characteristics now. And the general mentioned to you here now. The slime mold, to start with, they differ from the true fungi. They differ from the true fungi. Okay. In their phagotrophic nutrition.
Could you repeat that again, Professor?
Okay. Now, what it means, now, I ask question. You remember when, in the first, what is the way in which the fungi get nutrition? The general fungi, in which way they do the nutrition?
Generally, they are heterotrophs.
Heterotrophs, what do you mean heterotrophs? Heterotrophs, that means, just a minute here, don't confuse. Heterotrophs, that means if we divide all the organism on the planet, okay, I'm not going to go on region, we have two. The plant, we call them autotrophs. You know, autotrophs, that means they are able you see, they are able to, to produce their own food from a raw material. You know that. Because the plant, what happened here, they have they use water and carbon dioxide, and with the photosynthesis, they use it from raw material, and they can make and prepare from they prepare the starch. And that starch now is a prepared food, and the starch later on, as a glucose, and the glucose become a starch, and later on, all the other animals, including the fungi, the animals, these are all non-heterotrophs. Heterotrophs, that means they need a processed food already there. You follow my point of view? That is mean heterotrophs, okay? But the other point I want to mention, that enzyme, because they have a solid cell wall, so they cannot engulf the material inside. We as a human or animal, they eat, so they engulf the food inside. You got this point? Okay. Now, taking the food inside, of course, what you call it? Okay. Phagotrophic. Phagotrophic means engulfing the food. Okay. Now, fungi, the general fungi, no, because what happened here, the hive goes produce enzymes, and the enzyme try to degrade the macromolecules outside there, and they absorb it. You see the difference? Okay. And that's why I will mention to you here, they differ, means the slime mold differ from true fungi in their phagotrophic and nutrition, because they engulf the food inside. Okay, they differ from true fungi by their phagotrophic and nutrition. Okay, good. Now, is that clear now? Yes or no?
Yes.
That's good. So this will be phagotrophic, so they differ. Okay, so they differ from the true fungi in their phagotrophic and nutrition, and in that, we call their somatic parts. Do you know what I mean by somatic?
Body?
Say it again.
Is it body? Somatic parts.
Yeah, because any organism has two phases. The somatic is the phase in which they grow larger and they develop, or sometimes we call the vegetative phase. You got this point here? The other phase is the reproductive phase in which they reproduce. So somatic is what you call like the vegetative phase. Okay? So that's what we're saying. So that's what to make difference. So they differ from somatic fungi, differ from the true fungi in their phagotrophic and nutrition, okay? Of course, and in their somatic phase or their vegetative phase, they lack cell walls. They don't have cell wall. Lack cell wall. Okay, they lack cell wall. Okay. And to mention, and consisting only from protoplast, consisting only, okay, of protoplasts. Consisting only of protoplast. Okay. Now, as I mentioned, okay, they're protoplast bounded by a plasma membrane. Okay. So as I mentioned, they differ from the true fungi in their phagotrophic nutrition, and in that their somatic parts lack cell wall, consisting only of protoplast, bounded by a plasma membrane. Bounded by a plasma membrane. Because simply, if you look at it here, this is basically the protoplast. There is no special development, and only bounded by a plasma membrane. Okay. But of course, when they develop their spores, right? No. Their spores, however, okay, their spores, however, are each enveloped, means surrounded. Their spores, however, are each enveloped by a rigid cell wall, So there is, okay. Their spores, however, are each enveloped or surrounded by a rigid cell wall, and thus similar to those of the true fungi, You follow that? And that's why sometimes we call these organisms, we call them fungus animal organism. Okay? Because in their vegetative stage or somatic stage, they act like animals, but when they produce and they develop their spore is similar, what you call, to the fungi. Okay, so their spores, however, are each enveloped by a rigid, means solid, rigid cell wall, as similar to those of true fungi. Okay. As already I gave you some group. Okay, the group of slime mold, as I mentioned to you, we have, as already mentioned to you, we have here the myxomycetes, or commonly known as the Plasmodial slime molds, okay, or the other one, the class Acresiomycetes, or known as the Cellular slime mold. Those are different classes, but I'm giving you this other class here known as a Plasmodiophoramycetes, which are known as the endoparasitic slime molds. For this?
Prof, all the fungus make a starch?
No, I think if you remember when I mentioned to you that in the fungus cell wall, it is, now, you are asking me if the cell wall of the fungus is made from starch?
I'm just wondering, like, is it that they produce a starch, or is it just a specific type that they produce a starch?
No, the cell wall, you got my point of view here. A starch basically is a glucose. You follow that? You follow my point here?
Yes, yes.
Starch is basically glucose. Now, what is the cell wall of the plant? What do you call the cell wall of plant? Made from what?
Cellular.
Cellulose. That's good. Okay. Plant cell wall is cellulose. Sorry. Correct or not? Cellulose. Now, what is cellulose? Cellulose basically is made up, the main unit of it is, when I'm going to talk, hopefully when I'm going to talk about the physiology of the fungi, I'm going to explain it more in detail. Okay. Now, cellulose is made from glucose, but it is a polymer. Okay, a polymer. You follow my point of view? A polymer, and that polymer, if the glucose, we put like a unit like this, 6 unit here, the glucose could be made from 3,000 molecules of glucose or more, a very large kind, okay? But also this arrangement, it differs. So that basically that's important. So you don't say the cell wall is made from starch. No, the cell wall made from cellulose. You got this point here? Now, fungi, before they used to think, because they used to put them with the plant, Okay, because they consider fungi as non-photosynthetic plant. Because they think they have cellulose cell wall. But Edel realized the cell wall of fungi is mostly composed of glycogen. Though some of them, but the majority are from glycogen. You got this point here? Did I answer your question?
Yes, thank you.
You're okay, good. We'll tell that more in detail. So basically that's what, now. So now we're going to talk about what we call to start with the class. We'll start first with the class, Mycomycetes. Which is commonly known as a Plasmodial, slime molds. Okay, for that, known as slime mold. Plasmodial slime mold. Okay. Now these are basically okay. Now we're going to talk about this class here. At the beginning, we talk about general class of the slime mold as a whole. Okay. Now, just one thing I forget to mention to you in the general about the whole slime mold is important to mention this point for you here. Okay. Sorry, before you start this, I'm going to mention a few points. You put it with the general of the slime mold as a whole. You got my point of view here. Don't get mixed now. Because I didn't mention this point, it's important to mention to you here. Now, for most of its life, this is under the general of the slime mold here. Okay. For most of its life, a slime mold exists. Okay. As a thin, free living mass of protoplasm. Exists as a thin, free living mass of protoplasm. Okay. Did you follow this? And sometimes they are about several centimeters across. They don't usually some across. Okay. And sometimes it's several centimeters across. Now, usually the plasmodial, they have a very thin slime over it. Really, if you have it in the petri dish and here you look focus in the microscope, you can see that it has like a slime covering it. So that's why we'll show it here. It is slimy to touch. Okay, slimy to touch. And also to mention this point, and they can change form, and can change form. Okay. And creep slowly because they creep, okay? And they creep slowly over the substrate, and they creep slowly over the substrate. Okay, much like a giant amoeba. Much like a giant. Okay, yeah. Giant amoeba. That's what I want to put you here, okay? Now, did you follow this point I mentioned? So as it moves, means as it moves, that's how it feeds. It feeds itself, of course, by engulfing bacteria, by taking bacteria, by engulfing bacteria. It feeds by engulfing bacteria. And also to mention, and a tiny bit of organic matter, and a tiny bits of organic matter Did you follow this point here? Okay. Or do you want me to repeat something for you? All right. So basically, as I mentioned, they have protoplasm, and when it creep a little bit, then it feeds on bacteria and small organisms. So basically, like animal, in this case, engulfing the food particle. Now, but what happened here? Usually, after they, most of the time, after they move, then when they stop, then gradually they start developing these kind of small spores, which sometimes look very nice. It could be this shape, or it could be sometimes this shape or this shape here. Okay, just to mention this point now here. After a period of feeding and growth, after a period of feeding and growth, okay, the Plasmodium moves, the Plasmodium moves okay, into a drier, more exposed location. Okay, moves into a drier, more exposed location. Okay. Now, once it reaches an area more drier, because as you know, fungi, slime mold also they need a very heavy moisture. And if it happens for you sometimes, if you are walking in the woods, which is a rainy day or very wet, that if you can see maybe some of them growing in a small bark or something, they look like protoplasm. Okay. Now, but once they dry, move to area a little bit dry, then they stop creeping and they start producing their spores. So I'm going to mention this point here, as I mentioned. Okay. After a period of feeding and growth, the Plasmodium move into a drier, more exposed location, and here it gives rise to one or more fruiting body. And here it gives rise, or produce to one or more fruiting body. Okay. When you put fruiting body, that means spores. Okay.
Sorry, Professor, is this still under general characteristics?
Yeah, these are the general characteristics. No, apply to all. Yeah, because the idea I tried to mention this one here because I didn't mention that they are feeding on bacteria and other organisms, and when the the Plasmodium will dry a little bit, then will start producing the spores. You got the point of view here, just as a general of their what you call behavior. Now, still I'm under the general characteristic here, okay, and general automation. Because most myxomycetes or smalls are quite small. Okay, are quite small. Okay, though basically they could separate about a centimeter, but their height, basically as a height, they will never be more than basically 1 millimeter or 2 millimeter. Just to mention to you this point, most myxomycetes or to mention the slime molds are quite small, reaching no more than a millimeter or 2 in height. Okay. Now we talk about the class myxomycetes, which commonly known as the Plasmodial slime molds. Okay. Now, the Plasmodial slime molds are characterized. The Plasmodial slime molds are characterized. The same thing as by a free living. Okay. You know what means by free living? When we said an organism is a free living, that means it is not a parasite. It's not parasitic. You follow my point of view when I said free living. It's a free living, creeping, okay, multinucleate, okay, multinucleate mass of protoplasm. Okay. This is the multinucleate mass of protoplasm. Okay. And also this one, and usually, okay, enveloped, covered, sorry. Okay, by a slime sheath. Okay. With that, and totally, okay, devoid means lacking, devoid, especially this one, the Plasmodium, devoid of cell wall. Okay.
Can you repeat this sentence again?
Yeah, I said, what's it called? The Plasmodial slime mold, okay, are characterized, okay, by a free living, a creeping, multinucleate mass of protoplasm okay? Usually enveloped by a slime sheath and totally devoid or lacking, totally devoid of cell wall. You got that now? Okay. Now, to continue with this, and which become converted, and which becomes converted, become converted into one or more fruited bodies, bearing spores. Okay. And of course, when the spores, of course, these spores that you basically, as a way, try to pass the period of its unfavorable condition. But once these spores spread, of course, going to germinate into a new organism. You should know. Okay, just to mention here, which become converted into one or more fruiting body which bears the spore. Did you follow this here? Okay. Now, generally, the class Myxomycete or the Plasmodial slime mold, okay, we have a three group in this one here, okay. Morphologically, the Plasmodial slime mold shows three types, okay, of Plasmodium or Plasmodium. Okay. The three types, I'm going to mention the names here for you. All right. Let's see it now here. All right. One type is known as Fenero Plasmodium. Can you read it?
Yes.
Known as Fenero Plasmodium. Well, this is one type of the Plasmodium. One is known as Fenero Plasmodium. The other one known as Fenero Plasmodium or Plasmodium. Okay. And the third one is known as a Protoplasmodium. Okay, so these are the three types which you can see. The difference is where basically the regular one or the most common one, as I showed you here, okay, as you can see, this is the representative. As you can see here, basically they have the veins, and sometimes if look at differences, the veins is very obvious. You can see that the veins, and also they have granular and so on. Okay, and also they have that slimy sheet and so on. So basically, these are the pattern of the veins and the granules that sometimes occur more clearly than others, and based on that, we divide them into these types. So I'm going to see. So I'm going to talk about each of these types, basically, a very summarized way. Now, okay. Then I'm going to talk a little bit about the Fenero Plasmodium, which is the most one which is encountered in nature, and the one which most of the time you see. Okay, the Fenero Plasmodium. Okay, as already you can see here, you can see it, for example, that all right, they are characterized. The Fenero Plasmodium characterized, as you can see, by the protoplasmic veins. characterized by these protoplasmic veins, characterized by the conspicuous or clear, characterized by conspicuous protoplasmic veins. Okay. And as I said, we call streaming. Sometimes we call streaming, or sometimes we call shuttle streaming. Okay. Character in which streaming, which is streaming, be easily seen under the microscope. That means if this is the plasmodium like this, and these are the veins, I'm just give you an example, the veins like this. Really, if you look at the microscope, of course, you see these protoplasm like this inside, and it moves in one direction, this direction, or some it moves in direction. Okay, so the movement, you can see it's going and coming like this. Okay, this we call streaming. Okay, so that mentioned here. To repeat all these three sentences, the Fenero Plasmodium characterized by conspicuous protoplasmic vein in which streamings may be easily seen under the microscope. And also the other characteristic I mentioned, as you can see here in this yellow one, the presence of these granules. To make this point here, it is granular. It is granular. Okay. It is granular and is quite conspicuous, and it is quite conspicuous. Okay, even when it's small. Okay. Now to mention this point here, it forms large, gelatinous, reticulate, well, the reticulate will continue, a protoplasmic sheath. Okay, so as I mentioned, as it grows, form large, gelatinous, reticulate, protoplasmic sheaths. If it's that is how it cover, okay? And it may cover an area, as large as several feet square. Sometimes I think what happened here, I think in the 60s or in the 70s, and because some of these, they form like a gelatinous sheet, and of course, some of this grew about to come of the spore, especially in the species called like Gola, they develop a very large spore like this, okay? And it happens sometimes, I think it was maybe very humid away. When people came out in the morning, they saw their yard is full of these creeping animals with the large things there. So they're very scared, but in the end, it was very harmless things. Yeah, just to mention a few here, as it grows, it forms a large, gelatinous, reticulous protoplasmic sheet that may cover an area as large as several feet square. This is basically for the first one. characteristic of the first one, or the Fenero Plasmodium, and this one is the most common in nature. You follow this point here? Now we're going to move to the other type of this group here, which is known as Fenero Plasmodium. A Fenero means smaller, which is very similar with smaller, known as a Fenero or Atheno Plasmodium. Okay, Atheno Plasmodium. This one, as a matter of fact, is a non-granular. Non-granular means you cannot see the granules, has no granules. It is a non-granular. And very transparent. So this one is non-granular. The other point, then it is transparent or very transparent. Okay, very transparent. And also, as I mentioned to you, it does not have that slimy sheath on the surface. Okay, and are devoid. And it's vein, devoid of slime sheath. You got this point? Okay. And also this one is not very common in nature, not very easily you can find. Just put this point here. Okay. It's been, okay, devoid of slime. And it's difficult to detect in nature. It's difficult to detect in nature. Any question? Okay. Then we move to the third type of this plasmodium, which is basically the Protoplasmodium. Okay, the Protoplasmodium. Okay. The Protoplasmodium never grows larger than 1 millimeter in diameter. The Protoplasmodium, okay, never grows larger than 1 millimeter in diameter. Okay. The other point in this case also, it is granular in structure. It is granular in structure. And also, and has a slime sheath, Okay. Did you follow this point? Okay. But one thing I want to mention, basically, it has no veins as in the first one. The other point being, but it's not differentiated also, but it is not differentiated into veins. And the other point I'm going to mention here, okay? Remember we mentioned the first one, there is what you call the protoplasmic streaming, and most of the is going to occur in one direction. This group here, it has a protoplasmic streaming, but it is irregular. Just to mention to be, okay? It is not differentiated into veins, and its protoplasm streams irregularly. And its protoplasm streams irregularly. Is that clear? Okay. So these are basically, as I mentioned to you, a few lines for each one mentioned about the group. But the final thing I'm going to mention, of course, that the Fenero Plasmodium, the first one we described, is the most common in nature. Is that clear? Okay. Now I'm going to talk a little bit about the reproduction. Now, simply, of course, as you can see here, really, this is it gives you some picture here. This is black and white here, although it's a life cycle here. Basically, this organism that I shows to you, this is the Plasmodium. Basically, when it's going to creep here, okay, then after a while, creeping, of course, is going to produce, as a matter of fact, like a stalk like this. And here you have basically what you call the Plasmodium will develop like this. And when the spores are developed, as a matter of fact, of course, later on these spores can germinate and produce a new Plasmodium. These organisms, in spite of they are very tiny as well, also sometimes they may show some sort of sexual reproduction, but I'm going to talk in a very summarized way, very summarized way. Now, under the reproduction, normally, of course, as you mentioned, in general, when the protoplasm is creeping, that once it's creeping into area, for example, it has less food, as I mentioned, or become more drier, then in that case, the creeping will stop and start to bring what you call forming these spores. Now, when the food supply dwindles, okay, dwindles, or there is insufficient moisture, okay. Then in that case, the Plasmodium, okay, the Plasmodium, in this case, will crawls into an exposed surface. The Plasmodium crawls into an exposed surface. Okay. And then producing what you call, and they all mention that fruiting body is initiated, or okay, or spores is produced. Is that clear?
you repeat that once again, Professor?
As I mentioned to you, basically, when the food supply dwindles or there is insufficient moisture, the plasmodium crawls into an exposed surface and the spore formation is initiated. Is that clear now? The other point I mentioned, usually, as I mentioned, usually their spores, sometimes it is nice. Okay, just to mention this one here. Okay. Then Sporangia. Yeah, there is Sporangia. Some are complex and beautiful here. Some of them are really beautiful. I don't have it with me here, especially when you see this one here. And sometimes though it is here black and white, I think, okay. But some of them are really of a very nice color. Could be yellow, red, white, okay. But at the same time also beautiful, and also at the same time it is quite complex in nature, okay. And I think, okay, so basically that's what's point in this one here, all right. Now, to mention another point here, the Sporangia form cell wall. The Sporangia form cell wall. The Sporangia form cell wall, which is made of cellulose or chitin. Okay. Because if you look at the cell walls like this one here, do you see this properly? And maybe you have it. Maybe they give you this in the lab. Yes or no?
Yeah, they did.
Okay, yeah. So basically, what I'm trying to put out, usually okay, then you find this is basically outside. It's known as the peridium, which is the outside. It's basically the cell wall, which is as made from what you call either cellulose or made from chitin. Okay, and inside, of course, these small line as the capitulum, and these are the spores. Simple. So simply, I'm not going to go into here at the moment. I'm going to mention these spores are extreme. Sporangia form cell wall, which is made of cellulose or chitin. Okay. Around each nucleus. Around each nucleus. Okay. Now, the point I'm going to mention to you here is this here point that okay, important. Now, the organism is diploid, okay. When they develop these spores, their meiosis occur and the spores become haploid. To mention this point here. Okay. The sporangia form cell, as I mentioned, of cellulose or chitin. Okay, and within the sporangium, within the sporangium, haploid spores are formed by meiosis. Okay, are formed by meiosis. Is that clear now? Okay, so basically within the each the nucleus and basically each this is spore. Okay, so sporangia forms cell wall of cellulose or chitin around each nucleus, and within the sporangium, haploid spores means they have haploid chromosome number. Haploid spores are formed by meiosis. The last point in this regard here, going to now, these spores are extremely resistant. These spores are extremely resistant, okay, to environmental, to adverse environmental conditions. All right. Then now we're going to move to the class. Still have about 5 minutes or so to the class, which is the acresiomycetes. Okay, which we call them commonly known as the cellular slime molds. Okay. Very simply, this one here, what we call cellular, as you can see, and the cellular slime mold here, basically in the Plasmodium, you can see the cell. You follow my point of view, and that's why we call it cellular slime mold. Okay. Now, to make this one here, now cellular slime mold, the resemblance to Plasmodial slime mold, the resemblance, okay, the resemblance to the Plasmodial slime mold is superficial. Okay. And as I mentioned, they have much closer affinities with amoeba. They have much closer affinity with amoeba. Okay. I'm going to repeat these few lines. The cellular slime mold, okay, the cellular slime molds, their resemblance to the Plasmodial slime mold is superficial, and they have much closer affinity with amoeba. Okay. Now, basically, I'm going to talk a little bit about their behavior or their structure here to mention. Now, during their vegetative state or Plasmodial state, during their vegetative state, or, okay, as you can see here, the cells behave as a separate solitary organism during their vegetative stage, or, okay, the cells behave as a separate, each one by itself, separate solitary organism. Is that clear? Okay. Also continue with this, and also they creep over rotting logs or soil, and they creep over rotting logs or soil. Okay. Or sometimes they swim fresh, or sometimes they swim in fresh water. They swim in fresh water. And the same thing as the previous one, also they take bacteria and other small particles to feed on. Okay. As I mentioned to you here, ingesting bacteria and other particles of food. Okay, is that clear? Did you follow that? All right. Then a few lines I'm going to mention about their reproduction here. And of course, the best example I told you about it is the Dictyostelium discodium. You can see it here on the already I wrote it to you called discodium, Dictyostelium discodium, the best example we can use for the on this cellular slime mold. Now we talk a little bit about their reproduction. Okay. Simply to mention this regard, when because it's left 3 minutes because this one is going to stop. When moisture or food become inadequate, the cells send out chemical signals. When moisture or food becomes inadequate or less, then the cell send out chemical signals, okay, which is this one, known as cyclic AMP, cyclic AMP, okay, which is simply, I'm going to put it down here, cyclic adenosine monophosphate. Okay, called cyclic adenosine monophosphate. And once they see this signal, they gather almost in hundreds or thousands, okay, which causes them to aggregate, by the hundreds or thousands for reproduction. Okay, I'm going to stop now because I think this is with 1 minute going to close, so I don't like to shut on you like this. How do you find yourself in the lecture today? Are okay?
Very good.
Very
That's
good. You're excellent.
All right. Okay, good luck. Okay, have a good weekend.
You as well. Thank you.
Thank you.
Thank
Bye.
you.
Welcome.
Thank you.